Sorry for the break in transmission. It's due to network problem. Idris, how are you? Idris.
Yes, sir. Good afternoon, sir.
Afternoon. Your voice is clear as usual.
Yes, sir.
So network took me off, but thank God I was able to reconnect. So I was trying to
Sir.
review what we did last week so that we can enter this week. Is that okay?
Oh, okay, sir. I'm new. This is my first time joining.
Jesus. Your voice resembles one of our students. Okay. Well, that is okay.
Yes, sir. All right,
Okay.
sir.
Okay. Let me continue to finish the review of what we did last time. Then we're able to start this week.
Right, sir.
Have you joined the platform?
No, sir.
They should add you to the platform so that you can access the recording of our first lecture and our second lecture.
All right, sir.
Even this
I'll
lecture.
drop maybe I'll drop my number on the chat now.
You may even send it to me because I'm in that platform. I'm typing my phone number. Do you understand?
Okay, sir.
On the chat now, so that you can send it to my WhatsApp because this is my WhatsApp number. Then I'll add it.
All right, sir. Thank you, sir.
So 08037. I've just sent it. So you just look at the chat and then you copy it and make use of it. Then I'll add you when you send a hi to me. It's okay. But why don't you join for the past 2 weeks? This is the third week. Idris, why didn't you join for a long time?
Sir.
I say, why? Is it because of registration problem?
I just finished my registration. Is it on Monday? I just did my registration on Monday.
Okay. No wahala.
Yes, sir.
But we continue. So we also looked at mutation last week. And we said that mutation is when there is a change in the arrangement of genes on the chromosomes. We know that this gene or traits that transfer character from a mother to a child or from father to a child, they are arranged on the chromosomes, and each of them has a particular location. Just like as you sit on a bench, somebody is sitting number 1, somebody is sitting number 2, up to 7 people in a bench. So the position occupied by each person is now considered as a gene on that desk. That desk is a chromosome. Now you have position 1, you have position 2, up to position 7. So each gene is located at a particular position, and that position is called locus. So locus is the position of gene on the chromosome. And if there is a change, if for instance you leave somebody in seat number 1 and take the person to seat number 6, you have caused a change, and that is a mutation. The character will change, everything in there will change. So we have different types of mutation, which you will see when you go back and review what we did. So today we are going to look at, we didn't finish what we have last week. We didn't finish what we have. So, but then time went off. So we said that when we come back today, we are going to continue with that lecture and finish up. So today we are going to look at what we call polyploidy. We are going to look at what we call what? Polyploidy. Say it. I want you to say polyploidy.
Polyploid.
Polyploidy. Polyploidy.
Polyploid. Ploidy. Okay.
Yes, ploidy. Now, and the name poly means many. I hope you understand. So polyploidy is a term used to describe cells and organisms containing more than 2 paired sets of chromosomes. When an organism contains more than 2 paired set of chromosomes, then the condition is known as polyploidy. Because last week we also discussed it. For example, a human being has how many chromosomes? 46 chromosomes. And these 46 are in what? Pairs. They are in pairs. I hope you understand. So first pair, second pair, third pair, up to the number we have. So if for a particular reason there is a cell now contains more than 2 pairs of a set of chromosome, then the condition is known as what? Polyploidy. The condition is known as what? Polyploidy. And most eukaryotic cells are diploid. For example, human beings we are diploid because we contain 2 sets of chromosomes. I hope you understand. We have 2 sets of chromosomes in our body cells. So if you take a cell from the eye or head or leg or hand or any part of your body and check the number of chromosomes in that set, they are what? Diploid. That means they are 2 sets. Is that okay? But then a condition, you have a condition where we don't have this diploid. We don't have 2 sets. We only have 1 set or one set of chromosomes. You don't have the opposite one that make it diploid. Then that condition is called haploid. When you have 1 set of chromosomes instead of 2 sets of chromosomes. So that is called what? Haploid. That 2 sets of chromosomes is regarded as 2n. That is diploid number is 2n. But when you have haploid, which is 1 set, then we call it what? In bracket n. So it's not 2n, but what? N. So a condition where you have 3 sets, 4 sets, 5 sets, 6 sets, 8 sets, 10 sets, then we are talking of polyploidy. You are now talking of what? Polyploidy. So the normal one is haploid, which is n, or diploid, which is 2n. Any other thing outside this, then we call it what? Polyploid. I want to know whether you are following. I want to know
Yes,
whether
sir.
you are
I'm
following.
following.
Yeah. So poly means many. I hope you understand. Polyploid
Yes,
means
sir.
many sets. Other normal circumstances, you have what? Diploid, which is 2 sets. I hope you understand. Or haploid, which is just a single set. So when you don't have single set and you don't have double set and you go and have triple set, then you are talking that triple is polyploid. When they are 4, tetraploid, then you say it's polyploid. When they are 5, pentacloid, they are polyploid. And then you continue like that. That is what we want to discuss today. Are you following me? Later, we are going to look at the advantages and disadvantages of polyploidy. But let's just continue with discussing polyploidy. What are the types of polyploid? Types of polyploid. I've already told you that you may have 3 sets. When you have 3 sets, then you call it what? Triploid. Then you also have 4 sets, which is what? Tetraploid. You have what? 5 sets, which is what? Pentaploid. You have 6 sets, which is called what? Hexaploid. You may have 8 sets, which is what? Octaploid. You may have even 12 sets. That is what? The decaploid and etc. I hope you understand. Are you following me? Are you following me?
Yes, sir.
So, so you may have
Yes, sir.
triploid, which is what? 3 sets. Tetraploid, which is what? 4 sets. Pentaploid, which is what? Pentaploid, which is what? 5 sets. Hexaploid. You may also have hexaploid, which is 6 sets. You may have octaploid, which is 8 sets. You may have the decaploid, which is 12 sets of chromosomes. So these are the types of polyploid. I hope you are following. Touch shine. I know you have joined, but you are joining us after about almost half an hour. So we thank God I have rejoined again. So we still thank God for making us to join back. We are listening. We listed the different types of polyploid. One, triploid, which is 3 sets. Two, tetraploid, which is what, 4 sets of chromosomes. Pentaploid, 5 sets of chromosomes. Hexaploid, 6 sets of chromosomes. Octaploid, 8 sets of chromosomes. Dodecaploid, 12 sets of chromosomes. Example of a plant, plant that contains 3 sets of chromosomes is watermelon. They have 3 sets of chromosomes, so it's a polyploid. Then etc. like that. So you may not go to each and every one of them. So, but you can now understand what we mean by polyploid. Polyploid. You can now understand polyploid. That is when we are talking of different sets of chromosomes. Like the triploid is 3 sets of chromosomes. Tetraploid is 4 sets of chromosomes. Pentaploid, 5 sets of chromosomes. Hexaploid, 6 sets of chromosomes. Octaploid, 8 sets of chromosomes. Dodecaploid, 12 sets of chromosomes. Latif, are you, Idris, are you following? Tell me yes. Adenike, are you following? I want to get a feedback from you. Idris, I hope you are following.
Yes, sir.
Okay. Now, let us also continue. Let's look at polyploidy in plants. Polyploidy in plants. In fact, polyploid is obtained more in plants than in animals. It's very, very, very rampant in plants. One may ask why? Because when we go to the advantage of polyploid, you now see that plants that are polyploid, that have the duplex set of chromosomes, it makes the leaves to be better, or more leaves. It creates more leaves, creates more fruits, creates more branches, and it makes the plant to be robust. And then people prefer it. So polyploid can occur in plants, and the process begins if that diploid gametes are formed. And then the gamete may be formed by mitosis instead of meiosis. You know that gametes are always formed by meiosis. But when it forms by mitosis, it can form what? Polyploid. Do you understand? So it can occur in maize. Polyploid can occur in maize. It can occur in kernels. It can occur in other plants. So polyploid is very rampant in plants. So let us name some plants where there are polyploids. You'll be able to see polyploid in different plants, like domestic oats. Domestic oats, the number of chromosomes that's supposed to be in that cell is 7, but it can duplicate so that you have 6 sets of it. So you have 6n, which means 6 times 7, 42. So 42 is the chromosome number when you have a polyploid. Then peanuts, the original set of chromosomes is only 10 chromosomes, but you can form polyploid with 4 times the original number, that is 4n. So when you have a polyploid, then the number of chromosomes becomes 40. Sugarcane contains 10 chromosomes. I'm going to send all this to your to the platform so that you'll be able to see. I'm going to send the PowerPoint to your platform so that you'll be able to observe it and see what I'm talking about. So this sugarcane has 10 a set of chromosomes, but the polyploid form has what? 8 sets, 8 times 10. And that's why you have sugarcane that can grow big. Improve the variety because the chromosome number has been duplicated into 8 times. So the total number of chromosomes becomes 80 instead of 10. Banana is another example. it has duplicated. The total number of chromosomes in the original ancestral set is 11, but the diploid set now is, some are 2 times it, that is 2n, which is 22. Some are 3n, which is 33. White potato, the total number of chromosome is 12, but the diploid number is now 4 times 12, 4n, which is 48. Tobacco is 12, then it has duplicated into 4, that is 4 times 12, which is 48. Cotton is 13, then the polyploid form of it becomes 4 times 13, or 4n, which is 52. Apple, the original number of chromosomes is 17, but it has duplicated into 2 times. This is 2n, which is 34, and even 3n, which is 3 times 17, which is 51. So the polyploid in plants have larger cells and have larger fruits, larger leaves, larger branches, and etc. So that's why polyploid is very important in plants. But I want to ask you, does polyploid also exist in animals? Does polyploid occur in animals? The answer is yes. Agbaje, you are just joining now. You used to be the first person who joined the group. Agbaje, what happened to you today?
Yes, sir. I've checked it's like you sent the link late, sir. I check
Okay.
around 3.
Okay, thank you
So,
very much. Okay.
yes, sir.
Okay. So we have already revised what we did last week. And remember that what we are doing now is what we're supposed to finish last week, but we were not able to finish it. So we are carrying it over. I hope you understand. So it will push our lectures just like that. So, but what we are looking at today is polyploid. So polyploid is when you have more than 2 sets of chromosomes. Remember that the original number of chromosomes is either diploid, which is 2n, or haploid, which is n. Haploid is when you have only 1 set, but diploid is when you have 2 sets, as you have in every body cells. But then you can have 3 sets. 3 sets is polyploid. 4 sets is polyploid. 5 sets is polyploid. I hope you understand. As you can see, I was reading the number of chromosomes in various plants, and we were able to see that in domestic oats, the original number is 7, but it has duplicated. The polyploid number is 6 times 7, which is 42. Peanut is 10, but it has duplicated into polyploid is 4 times that 10. Sugarcane is 10, but the polyploid number is 8 times 10, which is 80. Banana has original number of 11, but it has become 2n or 22 or 3n 33. And so plant breeders used to manipulate so that the plant become polyploid, so that they can bear more fruits and have more leaves and becomes more cells becomes larger. So apple has the original number of chromosomes is 17, but it has duplicated to 2 times 17, which is 34, and 3 times 17, which is 51, etc. I hope you understand. That was what we listed. Then we now go and look at polyploid in animals. We want to ask, does polyploid also occur in animals? That is the question. Does polyploid, also duplication of chromosomes, does it also occur in animals? The answer is yes, especially in lower animals, especially in lower forms, such as, flatworms, leeches, shrimps, goldfish, salmon, salamanders, lizard. You can have polyploids in them. That means the number of chromosomes has been doubled. Instead of being a total of diploid or 2 sets, they have now exceeded 2 sets of chromosomes. So polyploid in lizard are parthenogenic. So polyploid lizard, when polyploid occur in lizard, then that lizard becomes parthenogenic. And what is parthenogenic? Parthenogenic is a condition whereby an organism or an egg hatches without fertilization. And when the egg hatches without fertilization, the animal becomes infertile. That means the animal becomes sterile. Even though the animal can grow, become big, but it cannot reproduce. It cannot produce. Fertilization cannot occur. So That is the danger in polyploid. So polyploid lasers are parthenogenic. And parthenogenic means that when they lay eggs, the eggs fertilize, the eggs hatch without fertilization. So a lizard doesn't need a female lizard doesn't need a male lizard if in that case. So that is polyploid lizard. It can also occur in other animals like insects, like reptiles, like amphibians. So polyploid can occur in some of these animals. So that, but one thing you should note is that whenever polyploid occur in animals, the tendency is that that animal becomes what? Parthenogenic. That means the egg hatches without what? Fertilization. And so it will be sterile. It cannot hatch. It cannot produce. It can grow to adult, but cannot fertilize another one. I hope you understand. So that is polyploid in animals. Then we now cross over to another subtopic, to the advantage of polyploid. What are the advantages of polyploid? What is the advantage? What are the advantage of polyploid? One, polyploid arise naturally. Sometimes you experimentally you can induce polyploid. Like you can use some colchicine, is a chemical. When you treat that seed with colchicine, it may becomes what? It becomes polyploid. Now, second advantage is that polyploid produce larger plant parts, e.g., larger leaves, larger flowers, larger fruits, and larger seeds. So if you want to get more fruits or more leaves or more seeds or more flowers, then you can make your plant to be what? Polyploid. When you induce polyploid, then you can produce larger leaves, larger flowers, larger fruits, and larger seeds. Then you can also resort to broader leaves. If you want to make the leaves to be broader, then you have to make the plant to be polyploid. Now, specialized characteristics such as seedlessness can also be produced. When your plants will not have seed. Some oranges that are produced as a result of crossing and polyploid doesn't have seeds. If you get your orange, you just lick it, no seed at all. So some oranges are like that, that's polyploid. They don't have seeds. Some pineapples are like that. They don't have seeds. But the danger is that it is very dangerous because that seedless plant cannot what? Produce another plant. Even though you enjoy it, it gives you more fruit, it gives you more seeds, it gives you more flowers, it gives you more fruit. But that plant is sterile. It cannot produce another one. I have an orange in my compound. That orange is seedless. But my worry is that I have been trying to get a plant of that plant, orange, but it's not possible for me. So what are the disadvantages of polyploid? What are the disadvantages of polyploid? One is sterility. I'm talking of sterile. Yes, sterility. You get the plant, it gives you what you want, but it's sterile. You cannot produce another plant. So sterility is a disadvantage. Some polyploids are unstable. Some polyploids are unstable. So they are not stable. Because, for example, if you plant it this year, it gives you fruit. Next year, the fruit changes. It is no longer as robust as the first harvest you made in the first year of planting. Like I have some pawpaw that are polyploids. It gives you big fruits in the first year. In the second year and third year, oh, what you see is baffling because the fruits become very small. So some polyploids are unstable. That is one disadvantage. An experimental introduction of polyploid is usually difficult. For you to experimentally produce polyploid plants is very difficult because they are unstable and most of them are sterile. So these are the disadvantage of polyploids. Even though polyploid is very good, it gives you larger flower, gives you larger fruit, gives you larger seeds, but then the problem, the disadvantage is that they are sterile, they are unstable, and experimentation with polyploid plants is always very difficult. To use it to carry out experiment is always very difficult. So let's now go to the next subtopic. The next subtopic is isolation mechanism. Isolation. What is isolation? Isolation is any external or internal barriers that prevent population from interbreeding. Anything that can stop population from interbreeding, you call it what? Isolation. It can be external barrier, it can be internal barrier. So any external or internal barriers that prevents what? Population from interbreeding, it is called what? Isolation. So when organisms are separated, gene flow, mutation, recombination, genetic drift, natural selection, etc. will not have effect on the other separated population. So you can now see any barrier that separates organisms that prevent them from interbreeding. You can now see that gene flow, mutation, recombination, genetic drift, they don't have effect on the other separated population. They may have an effect on the other one on this side, but they will not have effect on the other one on the other side. So isolation, as I said, is splitting the population of a species into separate units and the subsequent evolution into distinct species. So when isolation occurs, when that barrier, so the danger is that when they get separated, it becomes difficult. They now evolve differently. And so isolation is a very ecological factor that affects organisms. Now, what are the mechanisms of isolation? We said that isolation is a internal or external barrier that do what? Prevents organisms from what? Interbreeding. And so we may now look at mechanism. What are the mechanisms of isolation? One, geographical isolation. So the two types of mechanism of isolation is geographical isolation. Example, river. The river can separate two populations. Sea can separate two populations. Mountain can separate population. Desert is a form of isolation. It leads to isolation. Temperature, it can separate organisms. Forest can separate organisms. So these are what? Geographical. And they prevent what? Interbreeding physically. So when we talk of geographical isolation, you remember river, you remember sea, you remember mountain, you remember desert, you remember land, you remember temperature, you remember forest. Do you understand? And they are called allopathic separation because they force organisms to what? Evolve differently. So this geographical is also called allopathic. Then the second type of mechanism of isolation is reproductive isolation. Reproductive isolation. What happened here? Reproductive isolation occurs due to differences in genes that arise during the origin of such species that prevents interbreeding. So this one occurs when the organism is being formed due to mutation and other things that can make the organism to evolve differently. So this one is reproductive isolation. Reproductive isolation. Now, classification of this reproductive isolation. This reproductive isolation is more of internal, not external. Geographical is external, but reproductive is what? So we are looking at reproductive isolation that separates organism due to when the organism are being formed. So this reproductive isolation are divided into pre-mating mechanism and post-mating mechanism. Things that occur before mating and things that occur after mating. So let's look at pre-mating mechanism. something that occur before the mating. So this one is seasonal isolation. So potential mates do not occur together due to differences in breeding season. For example, toad. If the breeding season of male and female toads are different, it becomes difficult for fertilization to occur. This is also called a reproductive isolation because there is the variation in season, so that the male and female toad are not brought together, or male and female frog are not brought together due to difference in season. So it can cause reproductive isolation. So seasonal isolation can occur. These are pre-mating mechanisms. Another one is habitat isolation or ecological isolation. Here, potential mates do not meet each other due to differences in habitat, differences in space, differences in food requirements, differences in climatic condition, e.g. spawning ground differ in, make fishes in different tributaries. So what we are saying here is that habitat isolation is when there is differences in habitat, space, food requirements, climate, etc. It is also a reproductive word, isolation. And then behavioral isolation is also under reproductive behavior, behavioral or ethological isolation. Here, potential mates meet, but cannot mate due to differences in courtship behavior or signals. Here, the two animals may meet, actually, may come together, but they cannot mate because the courtship display are different. The courtship the courtship display are different, or even the signals are different that will result in the what? In the mating of the organism. So it is part of reproductive isolation. Let me summarize what we have done today. Summary of what we did today. Today we were able to review what we did last week, and I started today's lecture with polyploidy. And we said that polyploid is the term used to describe cells, an organism containing more than 2 sets of chromosomes. So when you have more than 2 sets of chromosomes, that is polyploid. So in that case, you have 3 sets. You can have 4 sets, 5 sets, 6 sets, 7 sets, etc. And then we have types of polyploid. One is triploid, that is 3 sets. Tetraploid, 4 sets of chromosomes. Pentaploid, 5 sets of chromosomes. Hexaploid, 6 sets of chromosomes. Octaploid, 8 sets of chromosomes. Dodecaploid, 12 sets of chromosomes. We'll give example when I send the slide to you in your platform. You see all the examples. They will also say that this polyploid occur in plants, and it is beneficial in plants because it gives the plant more leaves, more flowers, more